We will now discuss what is required for DNA replication, which materials are needed and necessary in order for DNA replication to proceed. We will also talk about the direction of replication. This is a very important point, recognizing the anti-parallel nature of DNA. You can imagine that this will create some problem for replicating both strands at the same time. So let's first start with requirements. So there are many different molecules and components that are required for DNA replication, but they can be grouped into three basic categories. First, DNA replication requires template. Secondly, it requires building material, it requires substrate. And thirdly, it requires enzyme and other protein which play a role in the synthesis of DNA strands. So what is a template? Template consists of a single strand of DNA. You remember during replications the hydrogen bonds are broken and both strands are released. Two strands are released and both can serve as a template for synthesis of new strands, semi-conservative replication. Raw materials are nucleotides. Basically, nucleotides needs to be added into the growing strand of DNA. So where do they come from? So the substrate which for nucleotides are nucleoside triphosphates. Enzyme such as DNA polymerases read the template, read the nucleotide and bring and add complementary nucleotide to it. During DNA synthesis, nucleotides are always added to 3' end right here. So the DNA replication proceeds from 5' to 3'. So the enzyme DNA polymerase, which adds new nucleotide, which actually recognizes a template nucleotide and brings complementary nucleotide, can only add this nucleotide if there is available hydroxyl group on the 3' end. So what are the substrates? What DNA polymerase recognizes? It is deoxyribose sugar, deoxyribonucleoside triphosphates. There are 4 of them: DATP, DTTP, DCTP, and DGTP. They are triphosphates. So once the DNA polymerase recognizes a template and complementary strand, it brings it to the close proximity of hydroxyl group of the growing strand of the DNA. Now this hydroxyl group attacks phosphate group on the nucleoside triphosphate and cleaves 2 phosphate out, and this one remaining phosphate forms a phosphodiester group here. So when this happens, the new nucleotide is added, and basically it is added to 3' end, and replication continues from 5 to 3'. Now, I want you to realize and not be confused when we said that replication always proceeds from 5 to 3' end, and that polymerase copies a template strand from 3 to 5' end. So realize this: we have DNA molecule. One goes from 5' to 3', and another, of course, we have anti-parallel DNA, so 5' to 3'. Now, this DNA molecule unwind and serve as a template. So the newly synthesized DNA strand will read the template from 3' end. So this would be 5' end of the newly synthesized strand, and it would grow towards 3'. So the synthesis of new strand of DNA go from 5' to 3'. However, it copies template strand here from 3' to 5'. The same here. So this is second template right here. So it would polymerase start at 3', which would be 5' of the new strand, and copy the template from 3 to 5' and synthesize a new DNA from 5 to 3'. So the synthesis of new strand of DNA always proceeds from 5 to 3'. However, it copies template from 3' to 5', and that's because of the anti-parallel nature of DNA. The anti-parallel nature of DNA introduces another problem. If you look at the replicon, let's pretend here is the origin of replication, so this is ORI, and replication fork is right here, which extends. So if you have two strands, so this strand of DNA goes from this way 5' to 3', and this original strand goes this way 5' to 3'. So basically what happened is that newly synthesized strand also has to go in opposite direction. So if this strand original is 5' to 3', so it would be copied from here. So the newly strand synthesized would go this way, and in this case you have 5' here, so it will be copied this way. So the synthesis on two templates of DNA is proceeding in opposite direction from the origin of replication. So how does this affect DNA synthesis? Let's look at the one side of the replicon. You see on one strand the DNA goes from 5 to 3', and another opposite way, of course, 5 to 3'. Now, as the DNA unwinds, the bottom strand, which goes from 5 to 3', continues, follows the replication forks. As replication fork unwinds, the strand is continuously synthesized, and we call this strand right here leading strand, and this synthesis is continuous DNA synthesis. Opposite strand you see on the top basically cannot go opposite way. As a fork unwind, it has to start a new synthesis. You know that DNA replication requires primers, so it has to add new primer and starts a new fragment to synthesize new DNA fragment. The same way it continues. Once the fork unwinds, new primers has to be added and a piece of DNA is synthesized. So this is so-called discontinuous DNA synthesis and create lagging strand. The bottom one was leading strand and top one is lagging strand. Now there is something a little confusing here, and it is that this picture only shows only half of the replication fork. And remember, when DNA unwinds, the replication bubble is created, so DNA starts to replicate. Afterwards, the more DNA is unwind, so replication fork extend and DNA synthesis continues. And it continues on both replication forks, so it is bidirectional. So what would happen if we outline whole replicon right here? So let's pretend this strand is 5 to 3', and this strand is 5' to 3'. Now here is replication, origin of replication. Replication start at replication fork at each origin, and it's bidirectional. So let me use a red one for the newly synthesized strand. So this strand will go 5' to 3' right here, so this will be 5 to 3'. And on the opposite template, this way it would be 5 to 3'. Okay, so this one would be 5' to 3'. Now, in this way, of course, it will go from 5 to 3', and here it will go from 5 to 3'. So you see it's bidirectional. So what happens when replication fork extends? So we have larger replication bubble. So let's say this would be our replication bubble now. So here is origin, so this strand was 5 prime, this is 3 prime, here is 5 prime and 3 prime. I apologize for my drawing. I am not really good in the drawing, but I hope that it will still help you. So now you recall in this way the 5 to 3 prime as replication fork unwinds can continue synthesis because it added primer, and as replication fork unwinds, the strand can continue synthesize from 5 to 3 prime. The same will apply for this strand on the second template. It can continue from 5 to 3 prime. So basically, did you realize that leading strands are found on both templates? What about second one? Here we had primer and 5 to 3 prime synthesis as replication fork unwinds. At this side of replication fork, on this template, new primer has to be added and a new piece of DNA is synthesized, so the Okazaki fragments are formed. The same apply for other side of bottom template. We had the first template, first the primer and first piece of DNA synthesized, which was 5 to 3 prime. So now as replication fork unwinds, a new primer has to be added, a new piece has to be synthesized. So did you notice something? That each template, bottom and top one, contains leading strands and lagging strands. The same apply for second one. You have leading strand and here are lagging strand. So it is not that one template give rise to leading strand and another template, second strand of DNA, gives rise of lagging strand, since two DNA molecules are anti-parallel. Replication starts at origin of replication and continue bidirectional, so it goes in both ways. Each template undergoes continuous and discontinuous replication. So on each template you find leading strand and lagging strands. So let's now summarize what we have discussed up to now. Let's compare the three models of replication: theta, rolling circle, and linear. Just by looking at the three pictures, I am sure that you immediately notice difference between rolling circle and other two models. What is striking immediately is that rolling circle is unidirectional and it creates a break in one DNA strand. It's unique to this mode of replication. So since rolling circle, the replication is unidirectional, it only creates leading strands, so it only undergoes continuous DNA synthesis. Now theta, even so, most of the bacteria, in most of bacteria which undergo theta replication, the replication is bidirectional. There are some which might be unidirectional. So in certain bacteria, the replication can be unidirectional. In eukaryotic replication, it's always bidirectional. So there are always two replication forks. Now, if you compare theta and eukaryotic replication, just look at the direction of replication. In both cases, replication starts at origin of replication and proceed at the replication forks. Now, in both cases, the template strand, each template strand gives rise to leading strand and lagging strands. Here, lagging and leading. The same in theta model. So of course we will discuss later on there are many differences between bacterial replication and eukaryotic replication. However, if you just look at the direction of replication as the leading lagging strain at the Okazaki fragment formation, continuous and discontinuous replication. There are many similarities in this regard between theta and linear eukaryotic replication.